Hello to our friends joining us via recording. Today we are talking about epithelial tissue and connective tissue. Before we started this recording, we did some group work together. So we're going to take some time to review some of the ideas from last week's lab that will help us with this week's lab. So let's start off with remembering when we talk about epithelial tissue that their names are really helpful for us to help us figure out what we're looking at. When we look at the names, we've got three different epithelial tissues here on the slide. So stratified cuboidal epithelium, simple squamous epithelium, and pseudo stratified columnar epithelium. When we talk about these three kinds of tissue here, their names tell us how many layers they have and what shape their cells are. So help me out in the chat here. When I'm looking at the first word in the names of these tissues here, so pseudo stratified, simple, stratified, what kind of information does that tell me about these tissues? Is that the layers or is that the shapes? What's my first word in each of these saying? Yeah, absolutely. The first word is the number of layers. And actually this first word that we see in each of these are the only three first words you'll ever see in epithelial tissue. So this is perfect. We get to review all of them. Let's start with our first one over on the left, the word stratified. Who can remind me what stratified means? Stratified epithelium. Yeah, exactly. So it's more than one layer. So it could be two or more. Whenever I see the word stratified, it just means that there's more than one. Absolutely, multiple layers. Some of the stratifieds we're going to look at today, we'll just see 2 layers. Some of the stratifieds we're going to look at today, we're going to see 50 layers. So stratified just always means there's more than one. Hey, which of my words over here, simple or pseudo stratified, which of those means there's only one? What's my word that means there's only one layer? Exactly. Yep. So simple means there's one layer. That word simple in its name means that there's just one layer of those cells. So simple tissues are going to be the thinnest tissues or the smallest tissues because there's just one layer of cells. And then here's my favorites over here, my personal favorite, the word. We got pseudo stratified over here. Yeah, Kaylin's absolutely right. Pseudo stratified. These are my fakers. I can't remember if I specifically said this last week, but I always love to say with pseudo stratified, these are the fake it till you make it types. So they only have one layer of cells. So when we're talking about their layers, they only actually have one layer of cells, but they're trying to make it look like there's more than one. So looks like more than one. Pseudo stratified columnar epithelium. Pseudo, remember from last week, means fake. Stratified means more than one layer. So fake, more than one layer, pseudo stratified. There's one layer of cells, it looks all funky. We'll look at a bunch of pseudo stratified today so you can tell, we'll know that we're looking at pseudo stratified because it's a hot mess. Faking it till you make it doesn't work for cells and it doesn't work for us either, right? It works to a certain extent until we just can't fake it anymore. And I don't know about you all, but with this pandemic, I'm about 2 inches from a hot mess. All right. Let's look at the second word in the names of these tissues. Oops, change colors here. So the second word in the names of these tissues is all about the shape. Absolutely, like Christian said, it's all about the shape. So we've got our 3 shape words here. When I talk about a cuboidal cell, how would you describe a cuboidal cell? This one should be the easiest for us, right? How do we describe cuboidal? Yeah, absolutely. Cuboidal, we look like a cube. So when you're looking at a cuboidal cell, it may not be totally cube shaped, but one of the big things you're going to see is in the middle, its nucleus is going to be really large, and it's also going to be about as long as it is tall. Cuboidal cells, our models make them look like little squares. They might not be quite be square shaped, but they'll be just as long as they are tall. If that's the shape we're looking at and they have a really big nucleus in the middle, that's a cuboidal cell. When we talk about squamous cells, how would you describe squamous cells? What were some of the things we said with our group mates there? Yeah, thin and flat, squish cells. Yeah, I love it. So when we first look at simple squamous epithelium, we're going to see a lot of cells that look like this. So it's really kind of long and skinny. It's really squashed or squished. The middle part is going to be where we find the nucleus. If you looked at simple squamous epithelium from the top down, which I don't think we have any slides that show this, but I'll just mention that a simple squamous cell from the top down kind of looks like a fried egg. So if we were looking at it from the top, this is what it would look like. It's really wide and expansive, weirdly shaped with the nucleus in the middle. Most of our squamous cells that we see are flat. They're squished. So we see those from the side. And our last one over here, yeah, right, Audrey said food shout out. I'm going to do all kinds of food shout outs. Maybe not as many as usual with tissues. Next week, we're going to be looking at steak, right? Because we're going to look at muscle tissue. So I'll ruin steak for you. All right, one last shape that we have over here, columnar cells. What are we looking at with columnar cells? What do those look like? Columnar cells. Yeah, Kaylin's totally right. Same thing with Elizabeth. Columnar cells look like columns. So columnar cells are going to have a nucleus down at the bottom. Here's a tissue word for you. The bottom side of a tissue, this is something called the basal surface, the basal surface. And the top side of a tissue is what we call the apical surface. The reason I mention these words is because this is going to matter when we're looking at stratified epithelium. When you look at stratified epithelium, there's more than one layer, do we look at the shape of the cells in the basal area or in the apical area to say what their shape is? If I've got more than one layer, do we look at the top or the bottom to figure out their shape? Yeah, a couple of us have chimed in. We always look at the apical surface, or we look at the top part of the tissue. It doesn't matter what's going on with the shape of all the cells that are down lower and kind of the basal area of the tissue. So I'll point that out when we're looking at our stratified tissues today. You're always going to look toward the apical surface. And with epithelial tissue, the apical surface is always going to be next to white on the slide, because the apical surface is the very outside of the tissue. And this is the kind of tissue that either is touching the air outside of your body, or the air inside your lungs, or it's touching your food in the digestive tract, for example. So the apical surface is always going to look white and empty. The basal surface is typically going to have stuff right next to it, connective tissue right next to it. So recap of what we learned or reviewed on this slide. We reminded ourselves of our layering words, simple, stratified, pseudo stratified. We reminded ourselves of our shape words, squamous, cuboidal, columnar. If you can describe those two things, that's a huge step toward identifying epithelial tissues. If you know what those words mean, that's going to help a lot. Let's take the words that we just talked about, and now we're going to do a little bit of matching. So I've got a list of types of epithelium that we see in cartoons over here. So let me label them with numbers so that it's easier to type. Get my T here. Okay, so number 1 on the top, number 2, number 3, and number 4. Let's start with you telling me which of my types of tissue that I see here would be considered simple epithelium. Which of my tissues here would be considered simple? Yeah, exactly. So remember from our last slide that simple means that there's one layer. So I have one layer of cells that I see right here. Notice that the way you determine the number of layers is where the nuclei are. So I've got one layer of cells. Notice that these cells are about as wide as they are tall. This picture makes them look really cube-shaped. They may not be quite as cube-shaped in real life, but tissue number 3 over here is an example of simple cuboidal epithelium. Simple cuboidal epithelium. When I talk about this tissue down here, this tissue down here is kind of a hot mess, right? I've got one row of cells, but unlike my pretty simple cuboidal here, see how those nuclei are kind of all over the place? They're going nuts everywhere. What was my word that meant I've got one layer of cells, but their nuclei are going nuts? They're all over the place. Yeah, exactly. These are those fake it till we make it's, right? So this right here is pseudo stratified columnar epithelium. Pseudo stratified columnar epithelium. These cells are tall and skinny, and their nuclei go all over the place. We will compare what simple columnar looks like to this pseudo stratified columnar in class today, so you can see how those things are different. Oh, I like it. Tien mentioned that when we talk about these pseudo stratified columnar epithelium, they kind of look like those blow-up dancing things that they put out in front of stores, right, or car dealerships. Although my favorite, I feel like, I guess pre-pandemic, whenever I was driving around, the place, honestly, that I saw those blow-up things the most was like e-cigarette stores. I feel like I was always seeing those wavy guys. Yeah, the wavy arm guys. I love it. So I feel like that's where I used to always see them. Yeah, mattress stores. So random places where they're really hoping that a dancing balloon is going to make you come. My dancing balloons, those pseudo stratified columnar epithelium. I love it. Perfect. All right, I've got 2 types of tissue left. These types of tissue have more than one layer. So I've got stratified is in their name here. Remind me with both of my stratified tissues here, am I looking at the bottom or the top to name their shape? Bottom or top? Where do we look? Yeah, absolutely, we're looking at the top. Now on these pictures, they were really kind in these sketches and they made the bottom and the top look the same. When we start looking at these things in tissues, they're not going to look the same in real life. So make sure you're looking at that top part. Up here in tissue number 1, I've got cells that are tall and skinny. What's my word for this shape here? Tall and skinny cells. Yeah, my tall and skinny cells, they look like columns. So this would be stratified columnar epithelium. They look like columns. Which means by process of elimination, that tissue number 2 is stratified squamous epithelium. Remember that squamous cells are really squished, so they're kind of long and skinny with a nucleus there in the middle. Stratified squamous epithelium. Now here's where we get into some new information. I mentioned this briefly, although I was cutting in and out, so you may not have heard me. There is a document posted for you called the Tissue Location and Function Guide. You should be able to find that in the week 4 folder. You can also find that if you're in the student Google Drive, that document is in there. Rather than going through and telling you all of the locations and all of the functions of all these tissues, which is super boring, we've compiled a document for you that has all of those locations and functions. There are more than what you need to learn on that list. You need to learn the ones that are in bold. So if you see any on that list that are not in bold, that's locations that might help you out in A&P2. But for the sake of A&P1, we are focusing on all of the things that are in bold. So the Tissue Location and Function Guide. This is a absolutely must-have document for this week, a reference document, because that's going to help you to know or to answer questions like this. I'm asking you to match up these 4 types of epithelial tissue with the specific location that they're found in the body. So there's some things in lab that are not good for flashcards. This is really good for flashcards. So build some flashcards, do it tonight. If you haven't already, make your flashcards tonight. What your flashcard should say is on the front side, the location of keratinized, stratified, squamous epithelium. That's on the front side. The location of keratinized, stratified, squamous epithelium. Who can tell me from my list over here, what would I write on the back? Where do we find keratinized, stratified, squamous epithelium? Yeah, Kira's right. Keratinized, stratified, squamous epithelium. We find that in the epidermis, or in easy words, well, easier words, right? In the superficial layer of the skin, the outermost layer of the skin. Next week, we're going to be talking about the skin more in depth. So we're going to look at keratinized, stratified, squamous epithelium this week and next week. Here's a little wink, wink, nudge, nudge. Since we're looking at this tissue twice, 2 times in lab, we better make sure we can identify it. Because I've got 2 weeks worth of questions when I'm building my exam that can include this tissue. So wink, wink, nudge, nudge. Anything that's found in the skin, whether it's my keratinized, stratified, squamous epithelium, whether it's my areolar, loose connective tissue that we'll talk about later, or my dense, irregular connective tissue, really make sure we know our skin stuff, because we're going to cover that 2 weeks in a row. I guarantee it's going to show up on the exam. So, keratinized, stratified, squamous epithelium. This is what you see when you look in the mirror. It's part of your epidermis, the outermost layer of your skin. Keratinized, stratified, squamous epithelium. This keratinized word means that it's full of a protein called keratin. We'll talk about this in lecture next unit. That makes it really tough or really resistant to damage. So that's good news, because when you're in contact with the environment, you want to have something tough there. Keratinized tissue. Yeah, it helps to protect you, absolutely, from getting hurt, from your skin tearing, all that jazz. What did this stratified word mean again? What was stratified? Yeah, so stratified was one of my layering words, right? More than one layer. So I've got more than one layer of really tough cells, that's keratinized, and the shape of those cells are my squished shape, right? My really flat cells. Keratinized, stratified, squamous epithelium. This is what covers the outside of your body. This is also the kind of tissue that you can thank for all of the dust in your house. Pretty much all of your dust is dead skin cells, and it's all this stuff right here. So keratinized, stratified, squamous epithelium, we find that on the epidermis, or the superficial outermost layer of your skin. Hey, let's talk about what we find another location of squamous cells in the body, and that's the alveoli, or that's the air sacs that you have in your lungs. When we talk about the alveoli, or the air sacs in your lungs, this is where we find simple squamous epithelium. Simple squamous epithelium. What was that simple word? What did that mean again? Simple epithelium? Yeah, exactly. Simple epithelium means there's just one layer. When we're talking about the tissue that we find in the lungs, where we need to get the oxygen from the air into your body, we don't want to have a ton of layers of epithelial tissue there. We don't want stratified. We want simple, because then that oxygen can come straight in from the environment into your bloodstream. So simple squamous epithelium, we find that in the air sacs, or the alveoli of the lungs. Let's talk about our next location right here. Our next location is the urinary bladder and the ureters. We didn't learn this structure in particular when we were working on organs, but the ureters are the little tubes that connect the kidney to the bladder. Here's the thing with those tubes and with the bladder. These are organs that can change shape depending on how recently you visited the restroom. So when it's been a while since we've been to the restroom, our bladder is going to be really stretched out. When it hasn't been that long, our bladder is going to be a lot smaller in size. When we talk about the type of tissue that we find here, we use what's called transitional epithelium. Transitional epithelium. Yeah, because like Kaylin mentioned in the chat, we've got to be able to change the size. We've got to go from really big to really small, back and forth your entire life. Transitional epithelium. And this is one that is probably my least favorite to identify because like its name tells us and like its location tells us, this is going to be changing shape. So I'm going to give you some hints about how to recognize transitional epithelium, but one of your big takeaways about it is that its shape transitions or it changes depending on how full these organs are in your body. So transitional epithelium in the urinary bladder and the ureters. That just leaves for us non-ciliated simple columnar epithelium. Hey, non-ciliated simple columnar epithelium is one of Dr. Ollis's favorites because look where it's found. It's found in my favorite system, lining the GI tract. So non-ciliated simple columnar epithelium. Columnar, remember, means they're tall and skinny cells. Simple means there's one layer. This non-ciliated thing, we talked about cilia last week. Yeah, non-ciliated, like Kira mentioned, that's going to mean that these cells have those things called microvilli. Microvilli. Can you remind me in the chat, what are microvilli good for? Why would a cell want to have microvilli? What's the benefit of that? Yeah, exactly. So microvilli help a cell to absorb nutrients, absolutely, absorb nutrients, because microvilli are actually folds of the plasma membrane. Imagine if you took a blanket and then you kind of folded it into loops up and down, up and down. That's what these microvilli are. So we take a big, large blanket and you squeeze it down into a third of its size, but you still got all that surface of the blanket to absorb nutrients. That's the goal of microvilli. So non-ciliated simple columnar epithelium. They don't have cilia, they have microvilli. I like to joke that we call them non-ciliated simple columnar because these are the younger brother of the ciliated simple columnar. If you are the younger sibling, inevitably when you went to the same school as them, right, they were always like, oh yeah, are you so-and-so's sister? Are you so-and-so's brother? So we're referring to this type of tissue like its older brother. It's ciliated simple columnar epithelium. We say it's non-ciliated because it's not the cool kid. It's not the ciliated kind. Remind me, before we move on here, if this was ciliated, what are cilia good at doing? If I did have cilia, do we remember what cilia are good for? Yeah, absolutely. Cilia are really good for movement. Movement of substances, I believe is how we technically said it last time. Cilia are going to help you move things like mucus or dust out of your respiratory tract. They're also going to be really important for moving eggs in the fallopian tube. So when they move from the ovaries down to the uterus, cilia help that movement to happen. So when we see our ciliated simple columnar epithelium, that's going to be in places like the respiratory tract where it's moving dust and mucus, or that's going to be in places like those fallopian tubes where it's moving eggs. So if you have cilia, you move stuff. If you don't have cilia, you absorb stuff. So another key word in their names to help us figure out what they're doing or where we might find them. One last thing that we did with our groups is we started to look at the functions of different types of tissue. And again, when we talk about these functions, remember that this is things you take from that tissue location and function guide. So make sure we find that in Blackboard, the tissue location and function guide, to make our flashcards, again, with our functions. So let's start up here. Our first function that we were matching up was the type of tissue that absorbs shock and it joins things together. Did we figure out with our group which kind of tissue absorbs shock and joins things together? Yeah, a couple of my groups nailed it. That is the job of something called fibrocartilage. Fibrocartilage. This is the type of cartilage that has a bunch of fibers in it, so we'll see that today. When we look at these two functions of fibrocartilage, that's going to help you remember what it does, or excuse me, where it's found. When we talk about absorbing shock, when we're talking about absorbing really hard movements or things like that, this is going to be the job of fibrocartilage in what we call the intervertebral discs. The intervertebral discs. That's the cartilage tissue between your spine, the bones of your spine. We've got cartilage there so that when you jump up and down, it doesn't crush your bones. That's the job of fibrocartilage. When we talk about joining structures together being a job of fibrocartilage, this is something that we see best in a structure called the pubic symphysis. The pubic symphysis. Hey, anybody want to take a guess based on its name? Where might we find the pubic symphysis? Yeah, Kira's totally right. We find it with, remember those bones in the hip called the pubic bones, the ones in the very front? Your hips are made of the ilium, where you put your hands on your hips, the ischium, where you sit, and the pubis in the very front. So there's some cartilage that holds together the two halves of the pelvis. That's fibrocartilage. It's really tough and really strong, and it makes up what's called the pubic symphysis. So the two functions of fibrocartilage, absorbing shock and joining things together, that can help you to remember where we find it in the body. Let's talk about our things that talk about pulling, our functions that talk about pulling. When we are learning the functions of a couple of our types of tissue, in particular, we're talking dense irregular connective tissue and dense regular connective tissue, their function is based on the way that their proteins are arranged in them. So this word dense in their name tells us that they're full of proteins. They're densely packed together with proteins. Sometimes those proteins are arranged regularly. And I'll show you this tissue. It looks beautiful. Everything's arranged in the same line. It's great. Sometimes they're not quite so regular. Sometimes they're arranged in every sort of direction. So when we look at this tissue, some of the proteins in it are going to be going left to right. Some of the proteins are going to be going up to down. Some of those proteins are going to be shooting out of your computer screen at you. Dense irregular connective tissue, the proteins are arranged irregularly. They're going all over the place. When we're talking about withstanding pulling or tension in one specific direction, do we think that would be done better by dense irregular or dense regular connective tissue? If we're doing one direction, we're always pulling in the same direction. Yeah, exactly. If we always pull the same way, we want all of our proteins to give us strength in that one direction. This is when, by the way, if I was cooler or maybe not cool. I would make some kind of joke about One Direction, but I apologize, I don't know any of their music well enough to be able to help to make that kind of joke. So when we're talking about dense, regular connective tissue, all of my proteins are arranged the same way. That makes me have a lot of strength in that one direction, that one way. So Alipina's totally right in our chat here. This is what we're going to find in things called tendons, which help us to attach a muscle to a bone, or it's also what we find in things called ligaments, which is what we use to connect two bones to each other. So tendons and ligaments, the two places where you find dense, regular connective tissue, where all of the proteins go in the same direction, making you really strong that way. When my proteins are going nuts and they're going left and right and up and down and forward and backwards, that's going to make me really strong in multiple different directions. Because if I pull you forward to backwards, you've got proteins there that do that. If I pull you up and down, you got proteins there that do that. So dense, irregular connective tissue is the type of connective tissue that's really good when we have pulling in multiple directions. Dense, irregular connective tissue is going to be another type of tissue that we see in the skin. So we see it in a part of the skin called the reticular layer of the dermis. The dermis, that's the second layer of the skin. So the lower part of this dermis layer, the second layer of the skin, has dense, irregular connective tissue. And think about this, you are typing on the keyboard, you are writing notes, you're playing with your phone. Hopefully not, but maybe. All of these things move your fingers in a lot of different directions. We're pulling in different directions. That's why I want to have irregularly arranged proteins, proteins that go all kinds of different directions. Dense, irregular connective tissue. That leaves for me my last type of tissue on this list, which is reticular connective tissue. Reticular connective tissue is another kind of tissue that I'm particularly fond of just because I like what its job is. So reticular connective tissue is a type of connective tissue. Think of it kind of like a net. So reticular connective tissue has proteins in them called reticular proteins. So those proteins, we'll do a little bad drawing here. Those proteins are going to have branches kind of all over the place. So by having these nets of proteins that are kind of all over the place, this type of tissue is really good at filtering things. So one of the locations of it, like my chat highlights, we're going to find it in the spleen. And the spleen is a place that's really important for filtering out old red blood cells or for filtering out microbes. So bacteria, viruses, those kinds of things. By having these proteins that branch all over the place, like we have in reticular connective tissue, we're really good at filtering things. We're really good at removing things. I spent a lot of time today going over these functions and locations to hopefully help us start making the connections between functions and locations. Again, flashcards are going to be super helpful for memorizing functions and locations. And the more you can try to make a connection between those two things, where it's found and what it does, the better off you are. Because, hey, check this out. We are wrapping up unit number 1, and we talked about the difference between anatomy and physiology. Here's a early review for us. Anatomy and physiology, they are or are not related to each other. Anatomy and physiology, they are or are not related. Yeah, they absolutely are, right? Remember the words we kind of used were they depend on each other or they dictate each other. When we're talking about the function of a type of tissue, would that be the tissue's anatomy or its physiology? The function of a tissue. Yeah, function is definitely physiology. So when we talk about these functions of our tissue, we're talking about what those tissues do. When we talk about where we find those tissues, that's one of our things that's anatomy, right? Where it's located, what it looks like. Where things are located, what they look like, gives us an idea of what they do, of what their function is. So as much as possible, try to pull together when you're studying these things. What do they look like? Where are they found? What do they do? Let's try to make those connections. Yeah, Kaylin's totally right. She said that physiology is going to make you have to apply things, and that definitely is much harder. I agree. That's why historically students always tell me, well, lab is way easier than lecture. It is, yep, because lab is mostly anatomy. Lab is, where is it? Can I identify it? I can memorize that. Physiology, I've got to apply it. I've got to understand it. So we're trying to tie together anatomy and physiology with these tissue types. If we can tie those things together, that'll help us a lot as we're trying to learn things. Before I move on, how are we feeling so far? Thumbs up, thumbs down. Do we have any questions before we move on? Somebody mentioned this earlier. I will keep the crowd happy here. It has been quite a while since we did a penguin. So while you're telling me how you feel, let me give you a penguin. Ooh, I'm a little out of practice. We're a little rusty. I got to get better at drawing them faster because you all finished your thumbs up before I got it done. Perfect. Okay. So what I'm going to do then is I'm going to switch screens on you, and we're going to start looking at our types of tissue together. So I'm going to kind of go in mostly the same order as is in your packet, not entirely, but we're going to follow it pretty closely. So let me give it a minute to catch up. Are we all seeing my first round of tissues on the screen here? Perfect. Okay, so we are starting here at the very beginning. The tissue that we are looking at here is simple squamous epithelium. So I've got two different views of simple squamous epithelium here. When we talk about simple squamous epithelium, remind me in the chat, what are the two things that its name tells us about this type of tissue? So we've got simple and squamous. What do those things mean for me? Yeah, so a lot of us are honing in. Squamous means they're flat, right? And simple means there's just one layer. So one layer of squished cells, that's what we're looking at here. Let's start over here on the right. Actually, before I dive in, a couple of us have mentioned in the chat the location that we find this tissue. This tissue is found, let me see if it'll let me write. Let's try. This tissue is found in the alveoli or the air sacs of the lungs. So when you're looking at this tissue here, these big open spaces that you're seeing on the slide, these are actually spaces that if this tissue was alive inside of you, there would be air inside here. So one of the first ways you're going to recognize simple squamous epithelium is you are going to see these big open spaces on these slides. Those big open spaces are where there would be oxygen. Simple squamous epithelium. These cells have one layer, or this tissue has one layer of cells, and those cells are really squished. So check out over here how I'm seeing this one line, and every so often in this line I'm seeing the little purple dots. Those little purple dots that you see are the nuclei of the individual cells. So in simple squamous epithelium, the dark spots that you see are the nuclei of the cells, and all of those cells are kind of arranged in one line. They overlap each other a little bit, but I have one line of cells. So in summary, simple squamous epithelium, how do we recognize it? Easiest first way to recognize it are these big white spaces that you see. Because remember, this is the tissue that's found in the air sacs of the lungs. Gotta have space for air. So number one, look for these big open spaces. Number two, when you see these big open spaces, look at the lines that form around them. If you see lots of darker dots in those lines, those dots are nuclei, which means I've got a bunch of teeny tiny cells that are wrapping around these open spaces. So simple squamous epithelium. Here's our first type of tissue. This is a type of tissue that some of us measured for the group project. What kind of tissue are we looking at here? Who recognizes this tissue? Yeah, it's mean for me to make you type tissue names, isn't it? They're very long. This is simple cuboidal epithelium. Yep, absolutely. Simple cuboidal epithelium. The way you're going to recognize simple cuboidal epithelium is you're looking for these little rings of cells. These little rings of cells are the cuboidal cells. So I can see those rings over here. They're a little bit larger on this one. They're a little bit smaller over here. Each of these rings are the simple cuboidal cells. Elapina's totally right. When we talk about where we find this kind of tissue, this is one of the types of tissues we find in the kidney. And these white spaces that we see here wouldn't be filled with oxygen. They would actually be filled with urine. So inside these white spaces that I see here is where we actually make urine. So we get rid of waste, we put that into the urine, we pull back water, we pull back nutrients, all that kind of stuff in these things called the kidney tubules. So each of these circles that you're looking at here has an open space in the middle. That open space in the middle is where I would make the urine. So on this slide over here, I'm seeing a lot of tubules, lots of these open spaces. Over here, I'm seeing more of the kind of connective tissue that's around them, but you can distinctly see that tissue right here. Remember when we talked about cuboidal cells that we said their nucleus was really big? So see these really big, wide nuclei that we see in these cells. And when you look at them, they're about as wide as they are tall. That was our definition of cuboidal cells. Notice in real life, they don't really look like cubes, right? But they're definitely about as wide as they are tall, cuboidal cells. Right now would be a good time for me to remind you all that part 3 of the lab group wiki project is available for you to complete. So plan to check that out after class today and maybe start working on that. It's one last application question from all the measurements that you and your group members took. So make sure, here's my wink, wink, nudge, nudge. Make sure when you're completing your set of questions, I'm either going to ask you for an epithelial tissue or a connective tissue. Make sure that you don't give me an epithelial tissue if your question's asking for a connective tissue, or vice versa. If your question asks for a connective tissue, don't tell me a type of epithelium. So make sure you're watching your words, take your time with it. That's the last part of the lab group wiki project, and then you're all done with that. So make sure we check that out after class today. That is due on Sunday, along with like everything else, right? Because we're coming up on the unit exam. So check out part 3 of the lab group wiki project. All right, so far we've looked at simple squamous epithelium, those one layer of flat squish cells. We've looked at simple cuboidal epithelium. Let's look at simple columnar and pseudo stratified columnar. So we've got our two examples of what column-shaped cells or columnar cells look like. Let me see how well I can write here. Over here on the left, yeah, Kaylin's absolutely right. Over here on the left, we've got non-ciliated simple columnar epithelium. Ooh. It's real tough when I have to write. Non-ciliated. Over here on the right, we have ciliated. Let's start first by talking about how you're going to identify if we are ciliated or non-ciliated. Check out this tissue over here. And yes, Elipine is correct. This one over here is pseudo stratified. Yeah, fake it till you make it over here on the right, this hot mess over here. The way we're going to identify that this is a type of ciliated tissue is if you look on the top of these cells, all these things up here that kind of look like eyelashes on the cell, all of those eyelashes are the cilia. And the cilia move back and forth, back and forth. They help to move the dust and debris out of your throat. They help to move the eggs down the fallopian tubes. Cilia, all about movement. If I'm moving stuff, I've got to be really long. So all of these little eyelashes that you see on top of these cells make this a type of ciliated tissue. When I come over here and I look at the top of these cells, notice how, by the way, my cells they're going all over the place. They're folding up and down, but there's only one row of nuclei. See how that dark purple dot is always kind of in the same place in this tissue? Because that dark purple dot, that nucleus is always in the same place, this is an example of simple columnar. It's not pseudo-stratified, it's just simple. Inside your digestive tract, you have little things that are called villi. So we talked about microvilli. Let's talk briefly about villi. Imagine if you hold your hand up and you see how your fingers kind of go up and down. So in between the fingers, it's down. On top of the fingers, it's up. So imagine that this is one finger and this is another finger, and then maybe here's your thumb sticking out over here. The tissue inside your digestive tract is arranged in little fingers called villi. So the reason that this tissue, we've got one layer of cells that goes up and down, up and down. It's going up and down because of those villi. So remember that non-ciliated simple columnar epithelium was what we found in the digestive tract. So here it is going all funky, up and down, because we're folding to absorb as many nutrients as possible. Notice on the top of these cells here how I don't have all of those long eyelashes. I just kind of have something that looks a little bit fuzzy. I kind of like to call this peach fuzz. We've got peach fuzz on the top of these cells instead of these really long eyelashes. That's what makes these cells non-ciliated. The fact that they've got peach fuzz on top of them instead of those long eyelashes. So when you look at a tissue, look for the top to see if we can see kind of a fuzzy looking appearance on the top where we have those microvilli, or do we have those long cilia that I see on the top up here? If you see those eyelashes, those long cilia, you call that a type of ciliated epithelium. If we see what's called technically the brush border, like Kaylin mentioned, or the microvilli, we call that non-ciliated. I want to pause for a moment and ask if we have any questions about identifying how the one on the left over here is different from the one on the right. Do we have any questions about telling the difference between these two? Or give me a thumbs up if you feel okay about it. It will take practice. So it's totally fair if you're like, I get it now, but I may not later. You'll definitely have to practice it. Okay, so I'm getting lots of thumbs up. We'll call that good for now. Remember that I will post this recording for you, and the guided lesson has more pictures as well. Here's the other thing I'll do, a quick shout out. Let me see if I can pull this off real fast. So I am a big fan of Google Images. I'm not sure how much I've used that in our class together, but I just want to pull over really fast. I did a quick Google Images search for ciliated pseudo-stratified columnar epithelium. Check this out. I've got tons and tons of pictures of this type of tissue. Now, you do have to be careful when you're looking at Google Images. Make sure that, like, if you were to go to the page that talks about this picture, make sure that it is actually talking about ciliated pseudostratified columnar epithelium, or, you know, simple cuboidal epithelium. But a really great way for you to keep studying these types of tissues is to look at Google Images, because you can find all kinds of pictures. Look at all of these pictures of ciliated pseudostratified columnar epithelium. All of them are going to have those eyelashes at the top, and all of them have that hot mess of nuclei going on all over the place. So a plug again for Google Images. Type in your search terms here. Here, we'll switch. Let's do simple cuboidal epithelium. Check this out. That looks a whole lot like what we were looking at before. Those circles around the open space in the middle. Or this one right here. Your teacher's going to nerd out on you. That's just so beautiful to me. I just love me some simple cuboidal epithelium. So check that out. Great resource for you, Google Images. All right, here's our columnars. We've done simple squamous, simple cuboidal, non-ciliated, simple columnar, ciliated pseudostratified columnar. What I don't have a picture of in this slideshow, because you have it in your packet already, is ciliated simple columnar epithelium. That's going to look very similar to this. It's just going to have eyelashes on top. So check for those eyelashes. Check to see if the nuclei are all in one row. If they're all in one row, that's simple. If they're all over the place, that's pseudostratified. And the ciliated or non-ciliated word comes from those eyelashes. Is it eyelashes or is it peach fuzz? That's how I get those words. All right, let's look at another example. Help me out in the chat. Which of these on the left or the right, which of these has cilia? Can we tell which picture has cilia? Left or the right? Yeah, absolutely. This is a great picture of those cilia. Look at all those little eyelashes on top there. So this is another example of ciliated pseudostratified columnar. I'll mention something too. Get out my pen. See this little thing right here and right here, right here. Ooh, that got funky. Okay, what you're seeing mixed in with this tissue here are a type of cells. You'll find information about this at the end of the lesson 4 packet. These things that I pointed to here, they're called goblet cells. Goblet cells are things that make mucus. So one of the main places that you see ciliated pseudostratified columnar epithelium is in the throat, so in the trachea. We make mucus in the trachea to catch any dirt or debris that we breathe in, to send it up and out. So you will see goblet cells in ciliated pseudostratified columnar epithelium. Some of the pictures that I have of non-ciliated simple columnar epithelium may also have those cells. Previous students told me the way they like to remember what goblet cells do, by the way. It's really gross, but here you go. Think of a goblet or a cup full of mucus. That is an image that will never leave our mind, right? Goblet cells. We got a cup full of mucus, and that's going to help these cells do their job and help us pass our anatomy exam, right? Goblet cells, full of mucus. Again, check out that sweet peach fuzz up here. Peach fuzz and not these little individual eyelashes. That makes this one over here non-ciliated. And again, you can see those little fingers, right? You can see those villi where it's folding up and down. So yeah, this would be the kind that we find in the GI tract. Really good at absorbing things. This peach fuzz is great at absorbing things. These cilia are great at moving things. All right, let's switch gears out of our simple epitheliums to start talking about our ones that have more than one layer. Remind me in the chat If you are an epithelial tissue that has more than one layer, what's the first word in your name if you have more than one layer? More than one layer. Absolutely right. We're starting to talk about stratified tissues. Stratified tissues. With stratified tissues, I'm going to ask the same question I've been asking all day. Stratified tissues, I look at the top or the bottom to figure out what shape the cells are. The top or the bottom when I'm figuring out their shape. Yeah, we're going to look at what's called the apical surface, or the top surface. Now that we're looking at entire tissues, it's a little bit easier to tell the bottom from the top, because the top part has this open space up here. Same thing over here, open space, connective tissue. So connective tissue is always going to look messier. Remember that connective tissue has a bunch of proteins in it. So we got a bunch of proteins going on down here. We got all of our cells that are epithelial tissue, and then we got open space. So you're going to look next to that open space to figure out what shape our cells are. These cells are really flat and squished. What would be the name I use for these cells? If they're really flat and squished, what was my shape word for these guys? Yeah, exactly. Squamous was my word that meant we were squashed or we were squished. I have more than one layer of cells. They are squished cells. These are what we call stratified squamous epithelium. Stratified squamous epithelium comes in two types, and it is important that we're able to identify the difference between the appearance of these two types. The first one that I have over here is what we call non-keratinized. Non-keratinized. All right, I never missed my whiteboard so much as I do today. Non-keratinized. What this word non-keratinized means is no keratin, non-keratinized. I have lots and lots of flat squish cells stacked on top of each other, but they do not have keratin. These ones over here also have lots of rows of cells, but notice how on the top I've got this one layer of cells that's like completely falling apart and falling off. This one over here is an example of keratinized, stratified squamous. Oh man, aren't you glad you're hearing me say it and not just trying to read it? Because that's pretty sad. Keratinized, stratified squamous epithelium. We talked about this earlier, and like Elapina mentioned, when we talk about keratinized stratified squamous epithelium, these cells out here that are falling off are completely full of keratin. They're very tough, they're very strong. They are also very, very dead. So when you're looking at keratinized stratified squamous epithelium, all of the cells in that outermost layer are going to be dead and they're going to be falling off. Compare that over here to my friends that are non-keratinized. They don't have keratin inside of them, so they are not dead. The cells on this outermost layer still look like squamous cells, and they're still alive. If you're not filled with keratin, you can stay alive when you live here on the outside. If you're filled with keratin, you cannot stay alive here on the outside. So here on the outside, look at those cells. If they're falling off, we'd say it's keratinized. If they're still all attached, we've got a flat line of cells. You can still see their nuclei out here. That's non-keratinized, stratified squamous. If you remember from the application activity at the end of last week, remember that we talked about how stratified epithelium is really good in places that are rubbing. So Obviously, with my keratinized stratified squamous epithelium, your skin is always rubbing against stuff. If we have multiple layers, we can lose some cells and we're totally fine. We find non-keratinized stratified squamous epithelium in places like the esophagus, for example. So here's a relevant example to recent history. Say you're having a Super Bowl party and you're serving nachos and you don't quite chew one of your chips just enough before you swallow it. The fact that you've got a ton of layers of squamous cells here gives you extra protection. We might scrape off a few of these top layers of squamous cells. We're going to have a whole bunch underneath them. Those whole bunch underneath them are going to keep your tissue protected. So stratified epitheliums, whether when we're talking specifically about squamous, stratified squamous epithelium, it's all about protecting your body. Keratin makes it even tougher, so tough protection. Non-keratinized is just going to protect you against rubbing or protect you against bacteria from invading. Both of these have multiple layers, and both of these have those squished cells. Stratified squamous. Who knows from the guided lesson what we're looking at right here? Does anyone know what we're looking at on these ones? Yeah, Kira's chiming in. She's correct. We are looking at Dr. Allis's least favorite kind of tissue to look at, or maybe one of my more least favorites in general. These are kind of the two main appearances of our tissues that have transitional epithelium on them. Transitional. Remind me in the chat, what did we say was special about transitional epithelium, about those cells? What's special about transitional epithelium? Yeah, absolutely. So I've got things in the chat like they're expanding or they're stretching, they're changing sizes. Totally. So when you look at transitional epithelium, my best hint for you when you're looking at it is if you see stuff that's like all kinds of shapes and you can't find one shape that seems like it's mainly what you got, you're probably looking at transitional epithelium. I had another colleague though that one of the ways she described it is this kind of tissue kind of looks like a cloud. So see here on the top how it's kind of bubbly and how these cells are kind of expanded and large. If we look over here, we've kind of got these big puffy cloud-like cells on the top. So the other way to think about transitional epithelium is we've got these big puffy cells. These are what the cells look like when your bladder is empty. So they're as big as possible. When your bladder is full, these are going to squeeze out more or straighten out more so that you have more space for urine. So when you're looking at cells that are arranged all kinds of funky, look at all this mess inside here. Who even knows what's happening? I look here at the top and it's kind of bouncing up and down. We got kind of a cloud puffy appearance down here. What we're looking at would be transitional epithelium. I don't have a picture here in my slides, but I will mention that you have in your packet some pictures of stratified columnar and stratified cuboidal epithelium. Remind me again what that word stratified means. What does stratified mean? Yeah, exactly. Stratified means I've got more than one layer. So remember that stratified cuboidal and stratified columnar, there is more than one row of these cells, whether they're column-shaped cells or whether they're cube-shaped cells. Stratified columnar, stratified cuboidal. With that, we transition into connective tissues. Now, let me mention, before we freak out, this one over here is actually a picture of epithelial tissue. I got a little funky on you. I got some connective tissue over here, but can anyone tell me what kind of epithelial tissue we're looking at in this picture over here? This one's epithelium. Yeah, Kira's right. This one over here on the left is simple squamous. Simple squamous epithelium. Yeah, and we're looking at our first connective tissue on the right, and this type of connective tissue over here is adipose tissue. Adipose. Okay, I want every single one of you to write this down somewhere on your lab packet or on where we're taking notes. This is what I need you to write down. I need you to write down, compare adipose, loose connective tissue, to simple squamous epithelium. I'll say it again. Compare adipose, loose connective tissue, to simple squamous epithelium. Because see, when I put these tissues side by side, notice how on both of these kinds of slides, it looks like I've got open spaces. We already talked about the open spaces here with simple squamous epithelium. Simple squamous epithelium, what would be in these open spaces here? What would I find inside here in simple squamous? Yeah, exactly. These are the places where we find air. So in simple squamous epithelium, you see open spaces. Those open spaces are the places where there'd be air in your lungs. When we look at adipose tissue, some of my adipose tissue slides look like there's open space as well. If we're actually alive, if this tissue was not just on a slide, yeah, these cells would be completely filled with lipids. Hey, we need to know this for next week's exam. There's a special name for these cells here in adipose tissue. I know it's a long thing to make you type. Do we remember what the name of the type of cells in adipose tissue is? Yeah, a couple of us are typing it, and it's a long word, right? We are looking at cells called adipocytes. Adipocytes. And in particular, like a couple of us mentioned, these adipocytes that we're looking at in these pictures, and in the pictures I'll show you here in a moment, these are examples of the adipocytes in white fat. Remember, we read the article about white fat and brown fat, and we need to be able to identify how those two are different. When we talk about white fat, remember that those cells had one big lipid droplet. So this big empty space that you see inside these cells, that's where the lipid droplet would have been if this cell was alive. When I put the tissue onto a slide, all of the lipids washed away. So it looks empty, but in real life, this would be full. There would be a huge lipid droplet inside of here. So white adipose tissue, it looks like there's an empty space. That's where the lipid droplet would have been. Simple squamous epithelium, it looks like there's empty space because there is. That's where the air would have been. One of the biggest key differences when you're trying to compare simple squamous to adipose tissue, notice how thin these little lines are here. These little lines that you're seeing here are actually representing the plasma membrane of each of these cells. So this whole space is just one cell. And for my friends who measured adipocytes, you know from the group work project, these are pretty big cells actually. Adipocytes can be pretty large. So this is one cell right here. This pink line that I'm looking at is its plasma membrane. If we compare that over here to this alveolus, to this air sac, there are tons and tons of cells that make up this line. There's the little purple dots that are the nuclei. I know they're kind of hard to see. You maybe see them better over here. So if you're looking at a wide circle and you see lots of little dots around on the outside, that's the nuclei. That tells you you've got simple squamous epithelium. If you look at this circle and it's a really thin line and there's maybe just like one place where you see a dark spot or one place where you see a dark spot, Literally, that's where the nucleus of these adipocytes got pushed all the way to the outside of the cell. They only exist to store energy, so all of this space is filled with lipids. They'll push those nuclei to the outside. So again, make sure we take plenty of time to practice when we're looking at our types of tissue. Compare adipocytes with a really thin membrane to our simple squamous epithelium, where those circles are made up of lots and lots of cells, so lots of nuclei that fill in this space. This is one of the ways that my adipose tissue slides will look. The other way that they will look is what we see over here. This view of adipose tissue, notice how my cells are orange. So I'm seeing these orange adipocytes. What's cool about this particular adipose tissue slide is that we didn't actually wash away the lipids that are inside these cells. So that's the orange that you're seeing here. We actually dyed the lipids inside of the cells orange so you could see them. So over here on the right, we see another example of adipose tissue where those lipids have been removed. They look empty on the inside. You can also see them though where those adipocytes look orange in color because we've actually dyed those lipids orange. So a couple of different ways that adipose tissue looks. Either we can see the lipids in the cells because they're stained, or those cells look empty like we see over here because those lipids came off. One other chance to practice here. I have simple squamous and adipose on this slide. Which half of my picture shows me adipose? Is it the left one or the right one that has adipose tissue? Where's the adipose? Yeah, exactly. Here's that adipose mixed in here. So I've got adipose tissue. See how I kind of call these cells, they're looking a little weepy. But at the same time, some of my alveoli look a little weepy too, right? But notice you can see those nuclei that are making up this alveolus. So I can see all the nuclei of all of the simple squamous cells that make up this open space. Over here, these open spaces are inside an individual cell. So please just make sure that we're comparing simple squamous epithelium to adipose connective tissue. That's one of those things that kind of get mixed up sometimes. Let's talk about, Can you still hear me? There we go. Okay. I think my computer froze for a moment there because it got stuck on the previous slide. I was like, oh, I hope I'm not dead. Okay. We got our next type of tissue up here. This type of tissue that we're looking at is what you call areolar loose connective tissue. Let's try to write it here. I'll try to write it down. Areolar loose connective tissue. That was a fail. Areolar loose connective tissue. The way that you're going to identify this tissue is you're going to see these dark spots that represent our cells, and I can see those cells over here as well. So the dark spots are the cells. They are a type of cell called fibroblasts. I'm not even going to try to write that and put it in the chat for you. Fibroblasts. That is another type of cell you'll find at the end of the lesson 4 packet. Fibroblasts are fiber builder cells. They build proteins. So when I'm looking at areolar connective tissue, you're going to see cells that are spaced out with lots of little proteins around them. So see how I've kind of got this protein right here that's kind of curled up on itself, or this one right here, it's a little curled up on itself. Those proteins are elastic proteins, so they make it really squishy inside the cell. There are also, I know it's probably hard to see on your screen, but there's some really light proteins here kind of in the background that branch a little bit. These really light proteins are those ones that we talked about previously called reticular proteins. So I've got some reticular branching proteins inside this tissue. I've got these curly elastic proteins, And then you're going to see some of the darker collagen proteins. That's the big thick lines that we see all over the place. Areolar connective tissue has all kinds of different proteins inside of it. So elastic proteins, reticular proteins, collagen proteins, they're all there. When I look up really close, so here's what it looks like with the yellow objective down, with low power objective. Here's what it would look like with high power objective, with blue down. So I've zoomed in a lot closer. Those fibroblasts look bigger. You can still see those proteins kind of branching all over the place. Notice though how there's still space on the slide. Everything's not completely filled up with proteins or with cells. That's areolar loose connective tissue. So 2 different colors for us to practice identifying. Sometimes the way we stain it, it looks blue, like I see over here. Sometimes it looks pink and purple, or pink and, I don't know, black-ish. My 2 types of areolar connective tissue. This is another one, by the way, that we're going to find in the skin. Yeah, so Elapina's correct. There's a part of the skin called the papillary layer. This is the layer that's right underneath the outside of your skin, right underneath the epidermis. So I'll show you at the end of our class today exactly where we find areolar loose connective tissue, but that's another one that we find in the skin. So make sure we can identify it and we know where it's found, because it's going to come up 2 weeks in a row. All right, we can identify this type of tissue. What are we looking at here? 2 pictures of something. What do we got here? Yeah, so Kira's naming the cells that we see here. Totally right. We are looking at a blood smear. So we're looking at my red blood cells called erythrocytes, and the white blood cells that come in multiple different colors. I got a red one over here, a really dark one over here. This might be a blue one. So we are looking at the white blood cells that are bigger. From the group work project, we should have figured that out too, that my white blood cells, my leukocytes, are bigger than my erythrocytes, that are the red ones here. When we talk about these leukocytes, or my white blood cells, what is their job? How might you describe what a leukocyte does compared to an erythrocyte? Yeah, I like it. Kira says leukocytes help to keep you healthy. Elapina says they help to fight an infection. Absolutely. So leukocytes, white blood cells, that's the immune system cells. Erythrocytes, red blood cells, those are the ones that transport oxygen. So most of your blood is made up of these erythrocytes, these red blood cells. Remember that we studied for lecture how water can go in and out. They're floating around in plasma. So the open space you see here is where the plasma would be. These little tiny dots that we're seeing here, do we happen to know what those little tiny dots are? What are those things called? Those little purple ones. Well, so the erythrocytes are the big red ones. This is probably an eosinophil. It's a type of white blood cell. These little tiny guys here, and you probably don't have to know them until AMP2, but I'll mention these little tiny guys here. These are platelets. These are platelets. Platelets are super small. These are the things that help your blood to clot. So platelets are these little tiny things that I see floating around next to those red blood cells. Platelets helping with blood clotting, those little tiny ones. So 3 kinds of cells in blood: erythrocytes, the red blood cells, the white blood cells that aren't actually white, but are kind of purple, blue, whatever, the leukocytes, the white blood cells, and then those tiny platelets. Yeah, you're totally fine, Kira. They're very small, hard to see, so you're totally cool. All right, let's talk about some of my dense connective tissues, When we talk about dense connective tissues, we've got dense regular and dense irregular. dense regular and dense irregular. So we got a vote over here for this being irregular and this being regular. Thumbs up or thumbs down? Do we agree? Irregular and regular. Yeah, lots of us agreeing correctly. This one over here is dense irregular connective tissue. This one over here is dense regular connective tissue. Remember that dense irregular connective tissue gets its name because the proteins are arranged irregularly. They're going all over the place. So some of these proteins are going up and down like this. Some of these proteins are sticking out of your computer screen straight at you. Some of them are going a little bit more sideways. Dense irregular connective tissue, again, I just like to say it looks like a hot mess. This is the kind of connective tissue that I like the least, probably, just because it's kind of ugly, like too much going on here. So dense irregular connective tissue, we already mentioned, is what we find in the other part of the skin. So this is below the areolar connective tissue. It's in the region called the reticular layer of the dermis, another thing in the skin. Remember though that this one is really good at pulling in all kinds of directions. So this is why it's good to have it around your fingers, for example, or under your skin, because we can pinch and pull and move in all kinds of directions. This kind of tissue is great for pulling in lots of directions. Over here is dense regular connective tissue, and dense regular connective tissue is a lot nicer to look at because it's arranged more regularly. So everything is going the same direction. These little tiny purple dots that you're seeing mixed in here, again, these are fibroblasts. These are my fiber building cells. So there's tons and tons of protein fibers that are inside this kind of tissue. That's why it's called dense. Both of these are dense types of tissue because there's so many proteins. Dense regular though, we see them in lines. Dense irregular, they are just all over the place. Yeah, and we will make our comparison, so make a note for yourself, and I know the guided lesson says this too, we got to compare dense regular connective tissue to smooth muscle. Next week we're going to do smooth muscle. Smooth muscle and dense regular connective tissue look very similar to each other, so we do need to make that comparison. For this week, we'll just compare these guys to each other, but coming next week, we will compare this to smooth muscle because that's another important one, another compare and contrast. Let's talk about my types of tissue that have purple in them. Here's something I want you to write down. This is another underline, highlight, star idea. Whenever you see purple, you should be thinking elastic. You should be thinking elastic. So anytime you see purple, we are thinking elastic proteins. So this is probably one of my favorite pictures that we have from our slides showing elastic. This is a dense elastic connective tissue. See these individual purple proteins that I'm seeing kind of all over the place mixed in with my pink collagen proteins. Dense irregular connective tissue had these pink collagen proteins too, but that was it. When I add elastic proteins to it, we call it dense elastic connective tissue. And when we have elastic proteins, that makes my tissues really elastic or really stretchy. So you'll find this in places like the aorta, which is the first tube that comes out of the heart. Your heart is constantly expanding and contracting, expanding and contracting. By having these elastic proteins there, that's going to allow that blood vessel to expand and contract to push blood through your body. So if you see pink and purple proteins, and we don't see big cells like we see over here, if you just see these pink and purple proteins, that's called dense elastic connective tissue. Over here, we're looking at those purple proteins with cells mixed in. The cells that I see mixed in here are called chondrocytes. And I'm going to type that in the chat because I don't want to try to write it. Chondrocytes. Yeah, so Audrey's totally right. A lot of times chondrocytes look like deer tracks. So see how they're coming in groups of 2? Chondrocytes like to live together. We will talk about this in lecture in the next unit. They live together in little spaces called lacuna. So anytime you see these groups of cells that are together with each other, we are looking at cells called chondrocytes. Chondrocytes are found in all my types of cartilage tissue. So if you see these chondrocytes and they're surrounded by purple, like I see here, you are looking at what's called elastic cartilage. Elastic cartilage. So here, let's recap. Anytime you see purple, we know that the name is going to have elastic in it. If you see purple with these big deer track cells, elastic cartilage. If you just see purple and pink, dense elastic connective tissue. If I still see those deer tracks, but I don't see purple, like you're looking at in this picture over here, I've got those deer tracks, but I don't have purple, this is an example of hyaline cartilage. Yeah, just like Elapina mentioned in the chat, this is hyaline cartilage. Hyaline cartilage is the kind of cartilage that the fetal skeleton is built out of. And then during fetal development and up through the first couple of years of life, this hyaline cartilage is turned into bone. The cells in here, again, we've still got those deer track cells that we're seeing in here, but we don't have the purple elastic fibers around them. So the big way you identify hyaline cartilage is it's going to look pink, and you're going to see those deer tracks, hyaline cartilage. If I've got those deer tracks and I've got purple, that's elastic cartilage. Because remember, anytime I see purple, it's always elastic. So elastic cartilage here on the right, hyaline cartilage here on the left. Both of them have those cells called chondrocytes, cartilage cells. One other kind of cartilage, and I'm going to be honest with you, you're probably most likely to recognize this one based on color. So as you're studying this one, really study the color. Yeah, I like this one too. It's really pretty. This is what we call fibrocartilage. Fibrocartilage. And it gets its name because it has protein fibers in it. It's not those purple elastic fibers. All of these blue fibers that you see here, these are actually collagen as well. But when we prepare a fibrocartilage slide, we stain them a different color. So all of the blue squigglies that I see over here are tough collagen proteins. All the blue squigglies I see over here are tough collagen proteins. All of these little pink spots that you see, because this is still cartilage, are chondrocytes. So we're still seeing those cells called chondrocytes, but this time they're surrounded by those collagen proteins. Fibrocartilage. Yeah, Kaylin's totally right. This is the strongest kind of cartilage because it has all those protein fibers in it. And remember that this is the kind of cartilage we find between the vertebrae in your spine, holding the two sides of your hips together. This is that tough cartilage. And yeah, Tian asked a good question about those chondrocytes, asked if they're found in all types of cartilage. The answer to that question is yes. Most of the time, so hyaline cartilage and elastic cartilage, those chondrocytes look like deer tracks. In fibrocartilage, we don't see them looking as much like those deer tracks, but you see them here in bright pink or kind of purple. Since it's still a cartilage tissue, they would still be called chondrocytes. Absolutely. Let's talk about the tissues that I turn my hyaline cartilage into. So hyaline cartilage, again, is what we make the fetal skeleton out of. We turn it into bone tissue as we're developing in utero and then after birth. So there are two kinds of bone tissue. The kind of bone tissue over here on the left that we see these open spaces in, this type of bone tissue is called spongy bone tissue. Over here on the right, we're looking at compact bone tissue. Compact bone tissue is probably like the most recognizable tissue we're going to look at ever. So I hope we all get really good at identifying compact bone tissue. Let's start with that one over here. When you look at compact bone tissue, you're going to see these big tree ring patterns that we see. These tree ring patterns are where we find the cells arranged in bone tissue. Kaylin's totally right. The cells inside this tissue are the small black dots that you see everywhere. They're called osteocytes. Osteocytes, that's my word for bone cells. Like any other kind of cell in the body, osteocytes need oxygen and nutrients. And that's going to come from these dark spots here in the middle that we call central canals. So if this tissue was alive, there would be blood vessels inside these big dark spots. So my osteocytes, my bone cells, surround these central canals in the tree rings to help us make this pattern where they'll actually share nutrients with each other. So when you're looking at compact bone tissue, everywhere on the slide is going to be filled with osteocytes. It's going to be filled with calcium, which in this case looks kind of light around here, and it's darker that I'm seeing closer to the blood vessel. That's what I see when I look at compact bone. When you look at spongy bone, like its name tells us, it kind of looks like a sponge. There's going to be some open space. So spongy bone tissue, sometimes that space looks more open. I can see a little bit of bone marrow inside here. That's the red stuff, is the bone marrow. In this case, the pink is actually the bone tissue. And we'll talk more about this in a couple of weeks, but when we're looking at spongy bone tissue, these little fingers that we see here are something called trabeculae. I'm going to type that in the chat. Trabeculae. That's a word that we'll come back to when we're doing lesson number 6 in lab. You probably can't see them super well because I know it's hard for me to see on my screen, but there are some little dark spots inside these trabeculae, and those dark spots are still the osteocytes. They're still the bone cells. Yeah, to Audrey's question that Kaylin answered for us, we call them osteocytes because osteo means bone. Let's see if we remember. We talked a lot about chondrocytes. What do we think chondro means? Osteo is bone. Yeah, chondro is cartilage. Absolutely. If we can start learning those prefixes now, that's going to help us out a bunch in the next unit and lecture. So chondro always means cartilage. Osteo always means bone. Yeah, fibro, like Audrey mentioned, fibro is going to mean fibers. Absolutely. Fibroblast literally means fiber builder. So you can get clues from a lot of the names of the cells that we're learning. They tell us what they look like, what they do, all that kind of stuff. Absolutely. So osteocytes in bone tissue, chondrocytes in cartilage tissue. And fibroblasts, we're going to find those in all those tissues that have a bunch of fibers. So areolar connective tissue, dense regular connective tissue. We can't really see them in dense irregular connective tissue, but they are also there as well. Anyone that's got a bunch of fibers, we're going to have some fibroblasts there too. Yeah, great connections to make. Wanted to show you that some of my images of spongy bone also look a little bit different. So this is one that looks similar to what we just looked at. You can see the bone marrow that's mixed in over here in some spaces. Sometimes you'll also see that bone marrow look a little bit darker in color. So in this case, the purple is my bone marrow, and you can see those trabeculae a little bit bigger. You can also maybe see the osteocytes a little bit bigger on this one. But Both of these are what my spongy bone, excuse me, my spongy bone looks like, with space in it for that bone marrow, and then the bony structure itself that's shown here in solid pink. I believe this is the last type of tissue I'm going to show you today. This is the type of tissue called reticular connective tissue. Reticular loose connective tissue. See how well I can do this. Reticular. Remember from our earlier discussion about areolar connective tissue that reticular connective tissue kind of looks like a net. So see all these little areas where you can kind of see this net or kind of bubbly looking appearance here? Those are my reticular proteins. So reticular proteins branch a whole bunch. They're really good at filtering things. What you're actually seeing too, these dark spots, by the way, on these images of, these are actually some of those leukocytes, some of those white blood cells. So this, remember, is the type of tissue that does a lot of filtering. We're pulling out bacteria, we're pulling out viruses. So when we look at this tissue, you're seeing those immune system cells that are sitting on top of these nets. The nets catch the bad stuff. The white blood cells that we see go through and eat it up. They do a process called phagocytosis. They eat it. So these are my two best pictures of what reticular connective tissue looks like. You can see those little nets that help us to catch stuff. Reticular connective tissue. I know a couple of us said that there were some issues with the sound. Hopefully we caught most of it. We were just talking about those net proteins here in reticular. All right, I am out of time. So here's the quick teaser I would do for us. We have looked at all of the connective tissues and all of the epithelial tissues in the body. Several of those types of tissues we could find in the skin. So remember that we had a type of tissue we found in the epidermis, and we had a type of tissue we found in the papillary layer of the dermis, and we had a type of tissue that we found in the reticular layer of the dermis. So going into next week, as we start talking about the skin, we're going to want to work on practicing identifying some of those types of tissue on images of the skin. So this hot mess down here, this is the reticular layer of the dermis. This is where we found the dense irregular connective tissue. Those proteins that are going nuts, going all over the place, we can see those here. Up here is where we found areolar loose connective tissue, where I don't have quite as many proteins as down below. We have a few more fibroblast cells mixed in. That's the areolar connective tissue up here. And then here on the very outside is where I have the stratified squamous epithelium. And let's see if we remember, these cells up here are falling off. Would I say that they are keratinized or non-keratinized since they're falling off? Yeah, I'm making you type a long word, right? Absolutely. We would say that these are keratinized because they're falling off because they're dead. They're full of keratin, they are dead. So keratinized, stratified, squamous epithelium. All of those tissue types in the skin. Here's another ugly version of skin. But what I want to point out is if you go down below the dense irregular connective tissue, we've got another type of tissue down here in a place called the hypodermis. Can anybody tell what kind of tissue is down here? Yeah, a couple of us are chiming into the chat. The hypodermis, the place underneath your skin, is where we find adipose tissue. And this is actually the place in the body where you find the most adipose tissue, is underneath your skin, ironically. So adipose loose connective tissue, you can actually find that in many, many different places in the body. So if you're looking at a slide and it looks like there's adipose tissue on that slide, in addition to what you're looking at, you're probably right. There probably is. Hey, so before I let you go, I just want to pop up. Oh, it's gone. Let me find it really fast. The picture that I put on our bulletin board for the week was this. We have now talked about many of the types of tissue that we see in this picture. So check this out. This is that type of tissue where the proteins go in all kinds of different directions. Any ideas what this pink stuff is here on the outside? Kira's voting dense irregular. Thumbs up, thumbs down. What do we think? Dense irregular? Dr. Ollis is going to vote. Dr. Ollis is voting yes. Yep. We've got dense irregular connective tissue out here, this pink stuff that's going nuts all over the place. When I look around here, I've got multiple layers of cells. What was that word for multiple layers of cells again? Stratified. Yep, so I've got some stratified epithelium in here. Hey, this inside here, we're going to talk about next class when we talk about the skin. All of these little bubbles that we're seeing inside here, this is what we call sebum or oil. So this is actually a cross-section of something called a sebaceous gland that makes the oil that goes onto your skin to keep your skin waterproof, to protect it. So the way that sebum secretion works is actually these cells make it and then they like die. So these dark spots you're seeing are actually like cells that are dead inside the oil inside your sebaceous glands. So we're looking at a cross-section of an oil gland in your skin. Don't we just love our oil glands, right? That's everybody's favorite, right? I'm saying that jokingly as someone who battled acne for quite a while. Oh, my sebaceous glands, not my friend. So with that, I'm going to close out our discussion of tissues so we can ponder the heart. Yeah, pretty amazing, right? That we can use what we looked at today to start interpreting pictures like that. So I got nothing else for today. Remember that for tomorrow's class, we're talking lecture. So please try to work through lecture number 4, lesson number 4, about active and passive transport. We're going to put together how active transport is involved in this process. We're going to learn a little about neurons as well. So work on that for tomorrow, 1 o'clock. Here again, we will chat through that stuff. I'm going to go ahead and shut off the recording, but if you have any last minute questions, I will leave my mic on for just a bit.